안녕하세요. 저는 유승범이라고 합니다. 이제부터 모든 발표를 영어로 진행하겠습니다. 하지만 후에 질문하실 때 한글로 질문하셔도 편하게 답을 해드릴 수 있습니다. So welcome to my talk, and I'll be presenting about my paper published in the Nature Communications in this early this year. The title of my paper is called Multicentric Tracking of Multiple Agent by Anterior Cingulate Cortex during Pursuit and Evasion. And this work is primarily done at the University of Minnesota. Okay, so let's just sit down and think about what is a fundamental behavior in the animal's life, including human behavior as well. If you look at the monkeys in the left panel, you will see them. They will be looking for their foods. Usually, we call this as a foraging. And the most important thing about the foraging is to understand what is the target, what is my food, and where is my target, and whether I can afford it or not. Human beings are also pretty much similar. If you go to a grocery store, you will be looking for some food in a specific location. So we can draw analogy between these two behaviors and learn a lot about from animals' behavior. During the fundamental behavior, there are a couple of very important information for success of foraging. One of the most important information is the spatial location of the obstacles and the landmarks. On the left, you will see antelope foraging for their grass. During their foraging, they need to understand where the huge trees are, or if some cases, other obstacles. In human foraging, it's pretty much the same. Like you have to understand where your correct food is. Like for example, you will be looking at some tomatoes. In that case, in this like bird's eye view, you will need to go into this location. So, in summary, what I'm trying to emphasize in here is the importance of the spatial information for their successful foraging. It becomes even more important when animal's target is actively moving in real time, corresponding to the animal's movement. In here, cheetah is chasing the gazelle, and gazelle is always trying to avoid. And they are definitely aware of a cheetah's movement, and they change the spatial location relative to cheetah. So, reiterating, now animal success, the fundamental behavior is foraging, and the most important information that I'm trying to emphasize across multiple slides are the spatial information. Two important spatial information while foraging hunting could be one, the first one is the allocentric information, and two is the egocentric information. So let me start with the egocentric information, which is elicited in the panel A. Egocentric information is the information relative to myself. So in this paper written by Ed Moser and Gary Buzzacki, says that the self-reference information or path integration information is referred as egocentric information. In contrast, what we can see the spatial information is like an absolute location of certain landmark. For example, you can always figure out south, north, east, west. In that cases, we have very absolute locations, and all the information, for example, Penn Station or Times Square to New York City is existing in certain kind of coordinate system. So these two informations are pretty much equivalently information in any types of foraging and hunting. So the traditional approach to understanding of the spatial information is typically done in the usually the rodent system, especially the rat. And the most of the target brain area was hippocampal place cells and the entorhinal cortex grid cells. So this is very popular, so I may not need any further explanation. But to briefly say is that there are some kind of cells that is actively firing at a certain location of the open arena in the rat's spatial navigation. And there is some kind of periodic firing within the space which forms a grid-like representation of the space, which we call as a grid cell. And these are awarded as the Nobel Prize in 2014. So that means like a lot of like sheer momentum goes into research of these topics. I start from some three important questions with respect to the studying spatial information. My first question in this paper is that whether the spatial information is unique in the hippocampus and entorhinal cortex. So people pay less attention to the spatial information in other brain areas. So we can reversely say that what if there is some other types of information in other brain area? And the second question is, does egocentric and allocentric spatial information require separate brain areas? Maybe we don't know. We may say there's coexistence of those both information in the same brain area. And the third question I ask in this question in the paper is that whether the spatial information is represented constantly regardless of other information. In this context, other information can be reward information or any other different information that you can find in the foraging or hunting. So keep in mind these three questions, and you will be finding the answers in these about these questions. So the task I designed for studying the questions that I try to answer is so-called I call the virtual foraging or virtual hunting. And I'll demonstrate the video that monkey is playing the game that I want. So here you can see the monkey is controlling the joystick, which moves the yellow avatar in the monitor space. Within this avatar, the monkey's goal is to catch the square target, which is moving interactively according to monkey's movement, and avoid the predator, which is listed as a triangle. So if you get caught by a predator, you have to wait from 2 to 10 seconds, depending on which predator it was. Often there is two targets that you can change. Always you can change your mind, because if you think it's not worth to pursue, as you can see, monkey changed the target. So this is trying to mimic the natural situation of the animals hunting without losing any interactivity or closed loopness of the hunting itself. So this is the behavior summary that I want to introduce. So as you have seen, the monkeys are controlling joystick in order to control the self-avatar. They have to catch prey and they have to avoid the predator. Very simple rule. You don't have to teach them very long time. That's just kind of huge benefit of this task over the other non-human primate task, because usually the training takes like half a year to 18 months to train a monkey to particular behavior, whereas this takes only 6 weeks to 8 weeks to train this monkey. So there's different colors over here, which corresponds to different speed, which means like easier, difficult, and amount of the reward. So if it is more difficult, then there will be more reward. That is pretty much the same in the predator. It comes from 2 seconds to 10 seconds of penalty, depending on the speed of the predator. The panel B explains the exemplary path that monkey and other agents are taking. You can see the emphasis in this paper is how complicated the animal's behavior and also the other agent's behavior. So the C is the path that monkey has taken within the session. It's about 560 trials, which one trial consists of either success or failure of the hunting. The failure can be either you get caught by predator, or you go over the timeout period, which means like monkey can only hunt 20 seconds. The orange points are with the points of capture the prey. So it's kind of like distributed all over the monitor space, which is a good sign that we have a lot of variance in the data. The panel D indicates the location of the spike in the dorsal anterior cingulate cortex. So it doesn't have it seems it doesn't have any like a very specific patterns in here, but you will see there's a lot of interesting pattern pops up when we analyze the data. The rest of the ENF is like about the behavior, the reaction time, and I should not say reaction time, but it's time to capture, and the percent of the choosing high prey as signature of they understand the task. So in sum, both monkey understand they have to pursue the prey with a higher reward. But because it's very interactive task and there's a lot of like underlying artificial intelligence controlling the other prey and predator, the reaction time pretty much remains flat. So now we know that monkeys are able to perform the task. Now we have to decide the brain area to record. So for my task, I decided to choose Brodmann area 24 in the monkey, which we call as a dorsal anterior cingulate cortex. I highlighted with the red rectangular in here. This ABC array is the area that I mainly analyze. Of course, a near future, there will be another data coming from analyzing FG and JHI, but I hope you can see with another webinar for this data as well. So in here, I recorded from 96 channels of floating microarray. That means I get a lot of repetition of neurons, but also a large number of neurons recorded simultaneously. Once we get the simultaneously recorded data of many neurons, often it's very hard to analyze those real-time data. But one analytic tool to overcome the limitation was called as a generalized linear model, which we call GLM. So in here, the basic pipeline of the GLM that I used is, let's say we have one time point of scenery of like for example, monkey and the prey. We can decompose that into different states. That states can be relative angle between the subject and the prey, relative distance between this prey and self, and also like absolute location in the space, absolute angle, absolute velocity of self movement. I specifically designated blue color for the relative information and red color for the absolute information. Once you decompose the scenery, you can call these as a kernel filter. So now you put kernel filter on the state information, and you will get a scalar value from each filters. Then you can just sum it, and then you can pass it through the nonlinearity. Once you pass nonlinearity, the reason of passing into nonlinearity is to secure non-zero values, and then you will get some single scalar value that converts into the Poisson distribution. And you sample from Poisson distribution, and you will get some number of spikes within given time point. So this is model spike that you get from this filter, and you compare that with the real spikes that I get from monkey's brain, and you find the maximum likelihood of information between these two. Of course, we cannot just fit it and say it is tuned. We need always do some kind of statistical check. And for that purpose, I use cross-validation. But the very simple thing is I just split the data into 10. I train with the 9 data, and I test with unseen data that is remaining. And how I find which variable it is tuned, I just add it, and then I see the likelihood of whose value increases. So in this example, there's only one variable. And if you have 2 variables, you will have better fit. The same as here, 2 to 3, 3 to 4. And 4 to 5, there's no more increase in the likelihood. So I just stop here, and I say that this neuron is tuned for the 4 variables. The panel on the right is showing the example of the fits. The black is the model fits, and the gray is the actual data from monkey's brain. So if you have a small number of variables, it doesn't fit very well. But if you increase the number of variables in here, their fits are pretty good, and it doesn't increase that much from here to here, meaning that 4 variables explain this data the most accurately without overfitting. Now let's see the example cells that I find in the empirically found, and also I found in the model. So the most upper row is the each individual cells that are tuned for the self position. So it's not very strictly like a place cell, which is like very specifically firing at the one point, or either it's not grid cell. It's like not like a periodic firing in here. But you can see a lot of cells are pretty much tuned in various positions of the space. And all the left hand here, which I highlight with my cursor, laser pointer, is the empirical data, which means from the monkey. And compared to that, if you see model data, let's compare this data with this data. Model is pretty much accurately capturing the empirical data. If you see peaks in here, it overlaps in here. Also peaks in here, overlaps in here. This is not limited to the self information, which is the monkey information, but this is also corresponding to whether monkeys are tuned for prey information. So prey informations are also very strongly tuned, and model captures it pretty correctly. This is not just about the certain positional information, but I would call other physical information. For example, the monkey's neurons are tightly tuned for the angle of self-movement. So self-movement, so if I'm moving towards the 180 degree, the neurons are firing the most actively. Now, if you see the empirical data compared to the model, it's pretty tight fit. So it's kind of like very impressive achievement of the model, not just for angle, but also the speed of self-movement. Again, going back to the initial introduction, I emphasize the importance of allocentric information and egocentric information. Previous two slides were dedicated to emphasize that dorsal anterior cingulate cortex is tuned for the allocentric spatial information. But also in the same brain area, it is all the neurons are also tuned for the egocentric spatial information. So egocentric space for this figure, I just displayed in very intuitive way. So if you think about the diameter of this circle is corresponding to the distance, and you will find the angle relative to a relative angle between myself or monkey and the prey, or monkey and the predator. So there are some neurons that you can see very interesting tuning. This neuron is always firing when the preys are at the right side of the monkey's avatar. And even it's more sharp tuning, it's only firing actively when it's in the, for example, like 1 o'clock degree. And the models are capturing these pretty nicely. And also there's, interestingly, the relative information about the monkey's avatar and the predator avatar is also showing some interesting selectivity towards it. So until now, what I have been, before that, let me go into the like population result, which is like showing that my previous results are not just a cherry picking result of nicely tuned neurons, but population in general, there's a large number of neurons that are tuned for the allocentric information, which I emphasize in the most left panel, and the also large population of number of neurons that are tuned for the egocentric information on the right panel. So I'm emphasizing here, saying that, okay, the entorhinal cortex and the hippocampus are not the only brain area that has a very specific tuning to the allocentric spatial information and the egocentric spatial information, but dorsal anterior cingulate cortex can be one of the candidate brain area that represents spatial information. It is not very surprising because entorhinal cortex has some anatomical connection with the dorsal anterior cingulate cortex. So maybe it can be confirmation of that proof of concept. But it's kind of like an amazing thing that we found this as a first time. The middle panel I specifically dedicated from my paper is saying that there are some neurons that are tuned not just for one specific information, but 6 of information, 5 of information, which means multiple allocentric or egocentric informations are represented in just one neuron. And this leads into my next part of the talk, which is saying that, oh, actually, is there any neurons that are selected for multiple information? And is that information statically represented if the other variables change over time? So for that purpose, I would like to briefly introduce the concept of the mixed selectivity. The briefly what mixed selectivity you can find is that, let's say you have one neuron that is tuned for a certain kind of direction or orientation. And usually you will find the tuning curve in here. But if you have like if you think the color information in here, red, is not related to any kind of tuning information, and you tested it, and you realize that actually the tuning changed depending on number of red dots in the screen, moving toward a certain particular direction. And that is called as a mixed selectivity. In more brief way, we can say that neurons are tuned for multiple variables. And specifically, we want to find that that is like a tuned for in nonlinear manner. So the types of mixed selectivity you can see in here can be multiple of them. Specifically, multiplication is that now my tuning curve change is tuning curve A multiplied by a certain number B. So it's kind of multiplicative operation in here. And it can be also broadening or displacement. There's many types of things that we can describe as mixed selectivity. People usually don't call addition as mixed selectivity, but also it's kind of like in the loose term, if you allow the linear mixed selectivity, they also count this as the mixed selectivity. In my paper, what I did was like, I looked for these two and how many neurons that from my harvested neuron shows either multiplication of the tuning or the addition of the tuning. So actually neurons are, interestingly, not just tuned for a single variable, but also shows high mixed selectivity in here. So this is like a tuning of self-position and self-direction. So self-direction have some tuning peaks over here. The light blue indicates when the monkeys are pursuing low reward prey. And when they are pursuing high reward prey, their tuning gets like sharper and the amplitude gets larger. But you can see both additive components in here and also the multiplicative components in here as well. This is same for the prey direction, other informations as well. And this is a population information. So of course, given time, I should skip the additive factor. But if you just look at the multiplicative factor more than one indicates that like there is some nonlinear mixed selectivity. So this is like slightly smaller than one, meaning that, oh, actually there's some shrink of the selectivity and p-value is very significant, proving that for the angular information, there is a mixed selectivity in the DACC. But we can also question, is the mixed selectivity only specific for the angular types of information? And I found with a different method, which proves that there is a mixed selectivity for the different types of information. So what I did in here is also same. I split the data into the case where monkeys are pursuing high reward prey and low reward prey. And the left one is always the low one, right one is always high, gray ones are randomly shuffled ones. And see whether the percent of neuron tuned for that specific variable, for example, self-position, changes over time. So if you shuffle by randomly, all of the data doesn't show significant difference between the two, and that is typically expected. I mean, that should be guaranteed. But let's say, let's pick the prey position as a kind of good example. So in prey position, if you have like probably left one is a higher reward one. If you're pursuing more rewarding prey, that means 40% of neurons are tuned for the prey position. Whereas if you're pursuing smaller value prey, there's only like 30% of neurons very significantly tuned to the prey position, which indicates DACC neurons are selective for reward in a mixed selective way. Mixed selective way. And I think that is kind of like emphasizing my third question, whether the tuning of the DACC neurons are constant regardless of other information. And I can definitely say no, it is not specific for it. It is not constant over time or other variable existence. So, obvious question people can ask while seeing my data is whether my neurons are tuned because of the gaze position. So what I did as a control analysis is try to see the percent of neuron tuned for a specific variable by adding gaze position as an extra variable to run the generalized linear model, or without gaze position. So one of the clear evidence is they don't change that much. That indicates my tuning is not due to the gaze position or simply the eye position. As we expected, the neuron tunings are not because of the gaze position, saying that, oh, self position doesn't change that much, prey position almost overlaps, predator position as well. This summary plot shows that, in conclusion, my results are not because of the eye position itself, but more of the state information, for example, prey, predator, or monkey's avatar information. To summarize, there were 3 questions I tried to answer with this paper. 1, is spatial information unique in the hippocampus and kernel cortex? No. Both spatial and kinematic information is also found at DACC. Second question I tried to answer was, does an egocentric and allocentric spatial information require separate brain areas? And we can find both allocentric and egocentric information in ACC, and it's more in the mixed selective manner. So the question is the spatial information represented constantly? No, it's mixed selective again, and it's like changing its tuning depending on the reward information that they currently have. So now, using the new paradigm, I have proved that maybe not just rat, maybe non-human primates are interesting system to study, and we can study many interesting phenomena by virtual hunting. So where my research should head towards, and I suggest a couple of interesting directions I want to pursue. 1, of course, the animals are not hunting alone by themselves. They also collectively hunt for one target. So we can expand the current pursuit task into multi-agent pursuit task. And also, often the hunters or the foragers encounters the camouflage. So we can also mimic that easily into my task paradigm. So all those kind of interesting questions, linking between the natural situation, understanding of brain activity, is expected to done in the future. So this is the end of my talk. I would like to make appreciation to all of my advisors and collaborators, especially Ben Hayden, who was like unconditionally supporting me to success this project, which was very, very risky project. So I would really appreciate his help. And also a lot of interesting comments and the interesting intellectual inspiration from the current PI at MIT, Mrda Zazayeri. And also the future direction is also coined by a lot of my students in their lab, my own lab, which already has recruited a lot of students and postdocs in the 성균관 University and Institution of Basic Science at the 성균관 University. And as I alluded at the last moment, my lab, ULab, is recruiting for postdocs, grad students, and interns who are interested in pursuing unorthodox task paradigm that is studied in the systems neuroscience. And if you're interested, please email to this address.